So here's where we ended our last video. We were talking about phospholipids, and I warned you that in this class, and not just for this exam, but you will hear me talk about the phospholipid bilayer a lot, because the phospholipid bilayer is kind of central to how life controls what's going on inside of it. Okay, phospholipid bilayer. A bilayer means two layers, and these phospholipids that we've been describing do naturally, when you put them into water, they form these structures that are 2 layers thick. 2 layers thick because each molecule is amphipathic. By the way, question number 13 on the chapter 2 part of the study guide is, why are they described as amphipathic? And they're described as amphipathic because they have one end, the heads, that are hydrophilic, and the other end, the tails, which I think look more like legs, which is hydrophobic. And it's just the fact that they are amphipathic that they will organize themselves in these entirely fluid yet entirely stable bilayers. And this bilayer, it is the beginning of the cell membrane. Cell membranes are also called plasma membranes. Those are really not different things. I used to think they were different, and I just didn't understand the difference, but they're the same. And this is going to be the starting point of what separates the living cell from the non-living environment, right? So we left off here, and we just need to conclude with there are other types of lipids, and one that we will be talking about, particularly when we get to the endocrine system, is this group called steroids, all right? Now, steroids are much more complicated than triglycerides. They still have a lot more carbons than they have oxygens, so you'll always be able to tell them by their formulas. But here's like carbon, 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 and you can see it forms a hexagon, 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 pentagon. That's the general structure of a steroid molecule. Why is it called a steroid molecule? I don't know. Chemists named it, so we'll have to ask them. But steroid hormones in the body are made with this basic structure. You can see this molecule here also has that same basic structure, and that molecule is cholesterol. You might know someone in your family who has cholesterol levels that are too high, and maybe they were surprised to learn that the cholesterol that's in their body is making. And they're like, why would it do that? Well, it's because cholesterol is actually a necessary molecule for life. It plays a role in the cell membrane that we're not going to talk about, but cholesterol is the starting point for synthesizing, which means making or building, synthesizing steroid hormones. And we will talk about, when we get to the endocrine system, the fact that steroid hormones are not just testosterone, the way people think, but they also include estrogen and progesterone, which are female reproductive hormones. Steroid hormones also include cortisol and aldosterone. So we'll be talking about that later. So far, in terms of biomolecules, we've talked about carbohydrates. We know the monomers are sugars. We've talked about fats, and we know that the building blocks, not exactly monomers, but the building blocks are fatty acids. When it comes to carbohydrates, we've learned the difference between monosaccharides, which is a single ring, and disaccharides, 2 rings of carbs put together. And when you put lots of monosaccharides together, you've created a complex carbohydrate, which an example would be a starch. And we know that in the human body, a good example of a starch is the glycogen that our body makes in order to have really short-term storage of energy. But we also learned that the best way for the human body to have long-term storage of energy is in the form of triglycerides. And that's because triglycerides, because they are hydrophobic, meaning they don't like water, they will store a lot of energy in a relatively small, light package, which makes it easier for a human to carry quite a bit of energy in the form of fat without being too heavy to walk around. And then we learned about phospholipids, and now we know what amphipathic means. And we also talked about steroids, an example of it being cholesterol, but also the steroid hormones. Now what? Let's talk about proteins. Proteins are my favoriteest biomolecules, right? Proteins are the best. Why are they so great? Whenever I read about how there's a scientist that is trying to develop nano robots to inject into the bloodstream to heal diseases, I always yell at the newspaper I'm reading or the TV. Proteins are nano robots. Actually, proteins, not all of them, but most of them are little tiny machines. And why are they like robots? Because they have got one job to do, and that job might be grabbing something and ripping it apart, like the enzyme that you will be working with in week 2, or grabbing 2 little things and putting them together. Now, we'll talk more about that. Right now, we're just going over the biomolecules, so we'll go over proteins in more detail. For right now, what you need to know is proteins are also organic compounds, biomolecules, and they are more complicated than the fats or the carbohydrates. And in order to make this more complicated structure, we needed more than just carbon, oxygen, and hydrogen. We also need nitrogen, right? All proteins contain nitrogen. There are some proteins that also have sulfur in them, but all of the building blocks for proteins contain carbon, oxygen, hydrogen, and nitrogen. Now, what do proteins do? I've said this before, and I'm going to say it again. Whenever we say that a cell does something or the human body does something like, the blood carries oxygen, it's a protein that carries it. Whenever we say that there is a cell that can protect the human body, it's because there are proteins that do something. If we say the liver can detoxify your blood, it's because there are proteins that do those things. Almost everything that gets done in the human body by a cell gets done by a little protein machine, right? And so that's a good reason to like them. So what are they? Let's see. They're very important structural materials. The strength of your bones is because of proteins in your bones. Your tendons and your ligaments are made out of protein. They are energy sources. That's not their primary role. But if we take in more protein than we need in a given day, your body will use that for energy, all right? There are some hormones that are basically protein hormones, like insulin. Insulin is the hormone that's related to sugar diabetes. That is actually a protein. The receptors on cell membranes, your cell has got that phospholipid bilayer, but it needs to know, hey, what's going on out there, right? What's going on in the environment? How does it know what's going on? It has proteins that sit in the cell membrane and kind of reach outward. And when a certain molecule binds to that protein, then that is the cell's way of knowing what's going on outside, right? Enzymes, as in lab 2, which are proteins that either rip things apart or put things together or reorganize things, right? All of that. Now, you should know that the building blocks of proteins are amino acids. Actually, I should have said the monomers that build up the polymers, which are proteins. The monomers are the amino acids. So protein, like a starch, could be thought of as like a bunch of beads on a string. And the beads, in the case of proteins, are amino acid. Each amino acid contains a carboxyl group on one side, COOH, an amino group on the other side, right? And this carboxyl group is attached to the amino acid, the amino group of that amino acid, and this amino group is attached to that carboxyl group of that amino acid. You know what? I got a picture of it in a second, okay? So if I'm an amino acid, I am one bead on the chain. The way I hook to the other beads are carboxyl group and amino group. And then I just have a hydrogen there. And then I have got hanging off the other side something that's called an R group. Amino acids are all the same except for their R group. And there are 20 different amino acids, which means there are 20 different R groups. I'm going to show you a picture. Okay. Now, what's the difference between a protein and a polypeptide and a peptide? All right. Okay. Listen to me because I'm a protein biochemist, so I know this stuff, right? Proteins, roughly speaking, are polypeptides. And peptides are just really, really, really short proteins. So when I was working in research at UCI Medical School, our field was protein and peptide biochemistry. And I asked my boss, who was teaching me everything, right? Well, like exactly at what length does a protein become a peptide or a polypeptide or a protein? And he was like, yeah, I don't know. It doesn't matter. So in general, though, peptides are really short proteins. Okay? So peptides are much more complicated than carbohydrates or the lipids. As much as I love phospholipids, proteins are much more cool. And so we needed nitrogen for that. So hydrogen, carbon, oxygen, pretty much every biomolecule add nitrogen in order to make the complexity that is an amino acid. Now, don't get afraid when you look at this. I am not going to make you memorize these structures or the names or anything about these amino acids. But this image is here because I want to explain something to you about protein structure. Okay, let's start with the basics. Proteins are polymers. They're made out of monomers. The monomers are amino acids. Roger that. Okay, there are 20 different amino acids in most of life. In most of life. What I mean is, if you're looking at a snail or a redwood tree or a mushroom or a lobster or an octopus or a human being, or most bacteria, it's just these 20 amino acids that are used to make all of their proteins. Now, there's one amino acid, this one here, that one there, that's found in some archaea, and we're not going to talk about it. Okay? But pretty much 20 different amino acids, no matter what form of life you are on planet Earth, at least. Now, in a laboratory, a scientist can make a lot more amino acids than this, but in real life, only these 20. Each amino acid's got a central carbon. Do we see that here? Yeah, okay. A central carbon is that guy right there. I know it doesn't say C because remember the abbreviation? Okay. And then it's attached to a carboxyl group that way, an amino group that way. It's just a hydrogen on the other end. And then whoop, it has got this thing, which is its R group. Now, the R groups, like that's an R group, that's an R group, that's an R group, they have got different qualities. Let's look at those, okay? Let's start with these guys. These guys here, these are 5 amino acids. Their R groups have got a charge. Now, I haven't told you this specifically yet, but I'm telling you now that any molecule that is an ion, which means it has a charge, either positive or negative, it does not dissolve well in lipid or in fat, but it does dissolve well in water. So these particular 5 amino acids, they are hydrophilic. They like to dissolve in water. And they have got charged side chains. These guys are positively charged. These guys are negatively charged. And you don't need to know their names, okay? Now, these guys, their side chains do not have a charge. They're not plus or minus. However, these guys are polar, so they still like to dissolve in water, right? So 1, 2, 3, 4, 5, that's 9 of them that like to dissolve in water. Now, let's look at these guys. These guys are hydrophobic. They do not like to dissolve in water. Now, as far as the side groups go, they can be very small side groups that don't like to dissolve in water, or really enormous side groups that don't dissolve in water, but they are hydrophobic. And then we've just got some extra ones in here, okay? Why am I telling you all of this? Wait, is it on here? Ooh, let's fix this, okay? So I'm telling you this because these amino acids, the way that they interact with each other is what creates the structure of a protein. I've been encouraging you to think about individual biomolecules, whether they are proteins or lipids or steroids, as being real three-dimensional structures that are as tangible in 3D as the chair that you're sitting on. When we're talking about proteins, and we're starting that now, it is particularly important. Here's one of the cool things about proteins. When proteins are being made, they do get made kind of like a beaded necklace. It'll be like one bead gets put on, and then another bead gets put on, and then another bead gets put on, and another bead gets put on, one amino acid at a time. And then as the necklace that's been built, as it leaves the machine that's building the necklace that's being built, it creates structure such that by the time the entire necklace is made, when it comes out the end of the machine that's building it, called a ribosome, it doesn't look like a necklace at all. It looks like a little robot. Maybe a little robot that grabs hydrogen peroxide and rips it apart, right? Or maybe a robot that grabs two things and puts them together. So it is built one little thing at a time, and as it leaves the machine that's building the necklace, it is going to actually turn into a three-dimensional thing that interacts with other molecules in its environment and moves and does stuff. It's not alive, but super cool, right? We're going to start there at the beginning of the next lecture.